So before we begin into DNA and how that works and how life is structured and that sort of thing, we do need to do a little bit of organic chemistry and talk about macromolecules. Macromolecules are the big molecules that are in your body. Macro means big. We're talking about very big molecules. I'm going to deviate a little bit from the course outline, and I'm going to talk about lipids, carbohydrates, and proteins. I'm going to leave our discussion of DNA to our next class and also do an online review because, of course, we do have the midterm coming up. Imagine we could build a cell from scratch, right from elements, and throw them together and make a cell. If we were going to do that, we need something to build the backbone of the macromolecules that form the structure of the cell and that form enzymes, etc. So what element should we use if we were going to do that? Well, we have to have an element that is capable of forming strong and lasting bonds, because there are some molecules in your cells that stick around for a long time and they need to add structural support, for instance. But we also have some molecules that need to be taken apart and reformed frequently. So ATP is used to shuttle energy around in the cell, and it can be taken apart into ADP and a phosphate, an inorganic phosphate, but then stuck back together right away. And it might be taken apart and put back together many, many times in a second. Also, if you look at DNA, DNA has to be unzipped. It consists of two strands, and they have to be pulled apart in order for your cell to read the information that's contained within the DNA, and then the DNA molecule has to be zipped back up. So we need some sort of atom that can form long-lasting covalent bonds, but also form weak interactions as well. We need an atom that can form many bonds, so it can stick itself to many other things, and that allows for the construction of complex molecules. We need to build some structures that are impervious to H2O, so they don't dissolve in water. But then we need to make some others that are soluble in water, because most of the chemistry of life happens in water. So we need molecular backbones that are versatile, interchangeable, reusable. They can be taken apart for energy production. They can be used for storing energy. They can be used for replication, etc., etc., etc. Carbon meets all of these requirements, and the reason for that is because it has 4 valence electrons. That makes carbon very versatile. So it only has 4 electrons in the outer shell. In order to be stable, it needs 8. And that means that carbon is capable of sharing 4 pairs of electrons with other atoms. If it's sharing 1 pair of electrons with another atom, that would be a single bond. If it's sharing 2 pairs, that would be a double bond. 3 pairs, that would be a triple bond, etc. But we can get some very, very complex molecules, any kind of configuration or shape you can imagine, using carbon. So all life that we know of is built around molecules that contain a carbon backbone. It's been hypothesized that silicone might fit this requirement as well, because silicone also has a valence of 4, and perhaps out there in the universe somewhere there is life that's quite different from what we know, and it's based around silicone instead of carbon, but we haven't encountered anything like that yet. So the study of molecules that have a carbon backbone is known as organic chemistry. Organic chemistry looks at molecules that are found in cells typically, although there are some that are associated with non-living processes also. And there's a whole naming system for organic molecules based on how many carbons they have, and also based on what other things are attached to that carbon backbone. So at the top here we've got methane, and this would be the simplest organic molecule. We've got 1 carbon and it's sharing 4 pairs of electrons with 4 hydrogen atoms. So it's got 4 single bonds, 1 to each of these 4 hydrogen atoms. And you can see the structural formula there. That's kind of the shorthand for this. That's the way you would draw this out. Again, a straight line, just one line means 1 shared pair of electrons. We could use a ball and stick model So if you've played around with chemistry sets in the lab, this is what you're doing. And then there's space filling models, which are a little more realistic because remember the orbits and so on are going to overlap. The next more complex organic molecule on this list here is ethane. And notice that we also have the formula, of course, that tells us how many atoms we have in the molecule. But notice that the backbone here is 2 carbons, and then we just have hydrogens attached to that. When we just have carbons and hydrogens like this, we call that a hydrocarbon, which makes sense. Now down the bottom there, we've got ethene or ethylene. And notice it's a bit different the way it's structured. So it has, again, just carbon and hydrogen. There's only 2 carbons, but this time we've got a double bond between the 2 carbons. So they're sharing 2 pairs of electrons. And that means that we have fewer hydrogens attached. We have 4 hydrogens instead of 6. I won't get into the naming system in great detail, but I do recommend that at some point you try to learn it and understand it because it will tell you a whole lot about the molecule. So for instance, meth, that first part of the name tells us that there's only 1 carbon involved. Eth, that tells us that there are 2 carbons. Pro, that tells us that there's 3 carbons, etc. The ane at the end tells us that there are no double bonds. There's only single bonds within the carbon backbone. The ene tells us there's a double bond within the carbon backbone. So these names all make sense and they tell you quite a lot about the molecule. So now that we've talked about some basic organic chemistry, let's go on and look at the actual macromolecules. And there's 4 varieties. We have lipids or fats. We have carbohydrates, which are sugars and also structural molecules as well. We have proteins and we have nucleic acids, which are RNA and DNA. A few terms that we should go through first. A polymer is a large molecule that consists of several building blocks, essentially. You can think of the building blocks almost like Lego blocks. Let's say you stick 100 Lego blocks together. You've got, I guess, a polymer of Lego. So polymers are made up of small building blocks, smaller organic molecules that are all very similar. The monomer is the actual building block. So for instance, proteins are long chains of amino acids. The amino acid would be the monomer. The protein itself is the polymer. And all polymers are made the same way in your cells. It doesn't matter if we're talking about proteins, nucleic acids. In all cases, the monomers come together and a water molecule is drawn off as a new bond is formed. So here we have an organic molecule of some sort, a macromolecule, and it consists of, to start, 3 monomers, 1, 2, 3. And we're going to add another monomer to lengthen this molecule. Note that at the end of the polymer, we have a free hydrogen. At the end of the monomer that's coming in, we have a free hydroxyl. They come together and a water molecule is pulled off and a bond is formed between the polymer and the new monomer. This is called dehydration synthesis. Synthesis because we're making a macromolecule, 1 piece at a time. Dehydration because we're pulling off water. If we want to break down a macromolecule, it's the exact opposite. We introduce a water and we break that bond that holds the monomers together. This is called hydrolysis. Hydro refers to water, lysis means to break. So the terms do actually make perfect sense. So now that we've gone over this, let's build a cell. So let's just imagine we're building a cell. We have these 4 different macromolecules. What role do those different macromolecules play? Remember that you are mostly water and your cells are mostly water, and all of the chemistry of life takes place in water. Water is essential. So we have cells that are made up of mostly water and they're surrounded by mostly water. So you have interstitial fluid between your cells. And your cell has to separate itself from its surroundings. Water is interesting, unusual, in that it's a polar molecule. Remember, that's because the electrons are shared unequally between oxygen and hydrogen, and that gives the molecule a dipole. It gives it a slight charge distribution that's uneven. And that means that water will grab onto anything that has a charge and it will solubilize it. So let's say we have a molecule that's got a slight positive charge to it. It's going to be attracted to the slightly negative oxygen of a water molecule, and then the hydrogens of that water molecule are going to interact with the oxygens of other water molecules, and basically that molecule is going to be dissolved or suspended in the water. So in order for the cell to not simply disintegrate in water, we need to have compounds that are not charged, that make up the membrane and make up basically the walls that are going to make up the compartments within the cell. And that's done by lipids. So lipids are formed by dehydration synthesis between glycerol and fatty acid. Lipids are kind of unusual compared to the other three macromolecules in that glycerol and fatty acid are quite different. If we look at proteins, like I mentioned, they're made out of amino acids that all have the same essential properties. But glycerol and fatty acid, those two components are a bit different. So we have glycerol, which is actually an alcohol. And if you look, we've got a chain of 3 carbons and we've got hydroxyls hanging off the end. Well, those hydroxyls can be used in dehydration synthesis to create a bond between the glycerol and a fatty acid. The fatty acid is a hydrocarbon. So you can see it's just a chain of carbons and hydrogens. Now remember that carbon and hydrogen have very, very similar electronegativities. And that means that the electrons are shared equally between the carbons in the chain and between the carbons and the hydrogens. So there isn't an unequal charge distribution, and that long fatty acid and the glycerol, after this dehydration synthesis has occurred, are hydrophobic. They don't interact with water because they don't have a charge. So this is a very stable molecule. And the one you're seeing here wouldn't be used to build a membrane. It would actually be used to store energy for long periods of time. So lipids consist of 2 parts. We have our glycerol, and then we have fatty acids, these long straight chains attached to that glycerol. Normally there's 2 or 3, but we can have variations of that. So we've got fatty acids that consist of a long carbon skeleton, usually 16 to 18, but that's quite variable as well. One end is a carboxyl group. We've got entirely non-polar carbon to hydrogen bonds and carbon to carbon bonds, of course, and that makes this fatty acid hydrophobic. The glycerol is also hydrophobic once it's entered into this arrangement with fatty acids. There are 2 broad categories of fatty acids. We can have some that are said to be saturated, and that's what you're seeing on the left, and we can have unsaturated. So saturated refers to the fact that that hydrocarbon contains as many hydrogens as it possibly can. So notice that the carbons kind of wiggle back and forth a little bit, but the chain overall is straight. It's just carbon and hydrogen. Now imagine that we introduce a double bond into the backbone of the fatty acid. In order to introduce a double bond, we have to lose 2 hydrogens. And you can see that the example here has 2 double bonds within the carbon backbone. And in each occurrence where we have a double bond, we're missing 2 hydrogens. What this does is it causes the chain to kink. It kind of upsets the electrons within the shell that are causing the double bond displace other electrons in that shell. We won't get into that in great detail, but just realize that it causes the chain to kink. So unsaturated fatty acids have kinks in them. We'll get back to saturated versus unsaturated in a bit, but first let's talk about triglycerides. Triglycerides are used to store energy long-term in your cells. There's other ways to store energy as well, but this is one of the most prevalent. So tri means 3. A triglyceride is a lipid that consists of a glycerol attached to 3 fatty acids. The fatty acids store a great deal of potential energy, and that's because all of the bonds within the fatty acid are non-covalent. And remember from our discussion of chemistry earlier, non-covalent bonds have a low bond energy. So those bonds between the hydrogen and the carbon and the carbon and the carbon are fairly easy to break. It doesn't take much energy. But when we combine the carbons and the oxygens that we pulled off with oxygen to make water and carbon dioxide, we get a lot more energy back. The picture that you're seeing there is showing fat droplets inside an adipose cell. An adipose cell is a fat cell. They actually start out very small, and as you store fat, they swell and swell and swell. So if you gain weight, you're actually not gaining cells, the cells are just expanding in size. Another really important group of lipids are the phospholipids, and as the name suggests, they have a phosphate group attached to them. We have 2 fatty acids, and they may be saturated or unsaturated, and they're attached to a glycerol. But then attached to that, we have something called a phosphate, and we have something else called a choline attached to that. The phosphate has a full negative charge, so it's got 1 extra unbalanced electron. And the choline has a full positive charge, so it's missing 1 whole electron. We have this region that consists of the glycerol, phosphate, and choline, and that's known as the head of the molecule, and it's hydrophilic, which means that it is attracted to water. And again, that's because water has a dipole and it will interact with those charges. The oxygen on a water molecule will bind to the choline, the hydrogens on a water molecule will bind to the phosphate. But the fatty acid region is hydrophobic because it doesn't have an uneven charge distribution anywhere. And down the bottom there towards the right, that's the way that this structure is usually represented and how you might draw it if you were to draw the membrane, for instance. That's where we see these lipids. So recall that a molecule that's hydrophilic is one that will interact strongly with water because it has a dipole or a full charge. A hydrophobic molecule is one that won't interact with water because it doesn't have an uneven charge distribution. An amphipathic molecule is a molecule that has both properties. So phospholipids are amphipathic. Amphi means both. Think of an amphibian. It can live in both the water and on the land. So phospholipids, the hydrophilic head will interact with water, the hydrophobic tail will not. So if you throw a bunch of phospholipids into water, they will rearrange themselves passively so that the hydrophilic heads are pointing towards the water and the hydrophobic tails are pointing away. It doesn't take any energy for these more stable configurations to form. So we can have what's called a micelle. That's where we get droplets forming, tiny little droplets. Or we can get a phospholipid bilayer. And as I mentioned, you can make your own phospholipid bilayer. You can make an artificial membrane by taking phospholipids, putting them onto the surface of water, floating them on top of water in a water bath, for instance, and vibrating it, shaking it. And some of the phospholipids will arrange themselves into this artificial membrane. Now, if it's an artificial membrane that you're going to use for research reasons or something like that, there's some other things you have to do to make it more resemble a membrane, but we'll get back to that in a bit. Okay, so as I've already alluded to, membranes are made up of phospholipids, and they form a barrier between the cell and the outside world, and they're also used to make compartments within the cell. So we have 2 layers of phospholipid. The inner core is made up of the hydrophobic tails, and the hydrophilic heads interact with water. And this is a very stable configuration. It doesn't require any energy to maintain that configuration. So because we've got a full positive and full negative charge on the heads, they are going to be attracted very strongly to water. And the tails aren't really being pushed away by the water, they're just being excluded by the water, and they're forming interactions with themselves. The tails the fatty acid tails, because they're quite long, there are actually van der Waals forces between adjacent tails on adjacent molecules that help hold that structure together. So this gives the cell protection from the external environment, and water, like I said, is actually potentially a destructive force. It will solubilize things, it will dissolve things. And also it allows the cell to make compartments inside, so things like the Golgi body, the endoplasmic reticulum, the nucleus, etc. On to our next macromolecule, and that would be sugar, carbohydrates. Sugar is also referred to as saccharin. It's a term that's not used very often in English, but let's say someone reads you a really sappy, sweet poem, you might say that poem was very saccharin. Anyway, a saccharide is a sugar. We can have polysaccharides, which consist of many sugars stuck together. But the actual sugar itself, the monomer, is generally 3 to 7 carbons in size, and usually it's a ring-shaped structure, as we'll see. So monosaccharides can exist in a straight or linear form, but generally they're bent into a ring. Notice on the first diagram there on the left, we've got the linear form, and notice that the carbons are numbered 1, 2, 3, 4, 5, 6, starting with the carboxyl. We won't talk about that in too much detail, but if you're wondering when you see these diagrams what the numbers mean, that's what they mean. So generally, monosaccharides are bent into a ring, and we call that a cyclic form. So we get bonds forming between the ends of the molecules, and in this case you can see that carbon number 6 is actually not part of that ring, it's outside of the ring. Down the bottom there, part B, you're seeing the abbreviated ring structure, and this is how biochemists and organic chemists would draw this structure. If you see an intersection there and it's got a number, you can see it doesn't actually have a C, it's just understood that at those points there's a carbon. Monosaccharides can come together through dehydration synthesis, just like other macromolecules, and we get a glycosidic linkage forming between the monomers. The numbers there, 1 to 4, 1 to 2, refer to the numbers of the carbons that are involved in that linkage. So here we've got the creation of a couple of disaccharides. Di means 2. And a lot of the carbohydrates that we consume as food are disaccharides. So sucrose down the bottom, that's table sugar. And it consists of a glucose monosaccharide and a fructose monosaccharide stuck together. Maltose consists of glucose and glucose stuck together. And in fact, if you break down starch, what happens in the intestines is it's broken down into maltose, and then maltose can be broken down further into glucose. Carbohydrates can be used to store energy in both plants and animals. In plants, starch is one of the main ways that a plant can store energy long term. So for instance, a potato is a highly modified root or tuber, and it contains mainly starch. And that starch is made up entirely of glucose. So we can have amylose or amylopectin. Those are two different forms of starch. Note that amylose is just kind of a spiral of glucose molecules. Amylopectin has branches in it. But each one of those green hexagons represents a glucose molecule. In animals, we store glucose as glycogen. And we do that mainly in the liver and in muscle cells. And glycogen is a highly branched molecule, but again, it consists entirely of glucose. And if your blood glucose levels drop, then that stimulates your liver to pull off those glucose molecules and dump them into the blood. And your muscles can store glucose for times when you need that extra energy. In many organisms, carbohydrates are also used for structure. Plants have a cell wall that's mostly cellulose, and that cellulose is a carbohydrate. It's made out of glucose. The glucose molecules are stuck together in such a way that we can't break them apart. We don't possess the enzyme needed to break the glycosidic linkages that are found in cellulose, even though we can break glycosidic linkages in many other molecules. So what you're seeing down the bottom here, B, we've got starch, and it's got this 1-to-4 linkage. And then in cellulose, on the right, we've got this 1-to-4 linkage as well. But notice that the glucose monomers are oriented differently. So you can see in the first diagram, B, the hydroxyl group that's highlighted there, they're all down the bottom. But in cellulose, they alternate. And that gives this glycosidic linkage a slightly different shape than we see in starch. And it means that the enzymes we have that break down starch can't do anything to cellulose. In fact, very few things can break down cellulose, even termites. They can't do it themselves. They have microbes that live in their gut that break these bonds for them. So cellulose is a very, very tough material. It makes up the wood that we find in trees. Even though there's lots of it out there, we can't consume it. Again, the large carbohydrates that are important to us in terms of nutrition are glycogen and starch. We can make glycogen ourselves, and we can break down starch that we get from plants. We have the enzymes to break both of these. Another interesting thing about cellulose is just how many hydrogen bonds it contains. So cellulose is formed by these long chains of glucose molecules, and again, the glucose molecule alters in its orientation. And that allows each glucose molecule to form hydrogen bonds with adjacent molecules in a different strand. So cellulose is a really tough, resistant substance. Really, really strong structurally. We can build houses out of it, but we can't eat it. But it's pretty amazing to think that it is glucose. I mean, if you go lick a tree, it doesn't taste sweet, but it's all glucose, forming this really, really tough wood. Some animals can take cellulose and break it down and get nutrition from it, but in all cases, at least all the ones I can think of, it's not actually the animal that's doing it, it is a microbe in its gut. So cows will eat lots of grass, contains lots of cellulose in the cell wall. They have a really complex stomach with several chambers, and they bring up the food as cud and they chew that and swallow it again. But despite all that, it's the microbes in their gut that are breaking down the cellulose for them. Another large carbohydrate that's difficult to break down is something known as chitin. Incidentally, if you see CH at the beginning of a word, and the word is derived from Greek, it's pronounced as a hard K, just something you might run into. Chitin is something that's produced by a lot of invertebrates that have an exoskeleton. So they have their skeleton on the outside, basically muscles attached to that skeleton. What you're seeing in B there is a really interesting insect, and I know I'm going off on a tangent again, but this is something called a cicada. It spends its entire life, sometimes decades, underground in the dark, sucking sap from tree roots, and then it climbs up. For one glorious month, it spreads its wings, it flies around and sings. It's the insect that makes that really loud buzzing noise in the trees that you might have heard. In any case, you can see its old exoskeleton there that it's shedding, and that exoskeleton is made of chitin. And chitin has some medical uses. It's actually very, very strong. It doesn't break down immediately, and it can be used to make surgical thread, for instance. So chitin is very similar to cellulose. We won't get into the details all that much, but just note that it's got some nitrogen in it as well. Cellulose doesn't have that, and there's not quite as much hydrogen bonding. Okay, so now our cell has compartments, and it's protected from the outer environment by the phospholipids. It also has something to eat. It's got food or energy for long-term storage in the form of triglycerides. If we're talking about a plant cell, we have a tough wall around the outside that's made of cellulose. We're not plants, but that cellulose is important to us because a lot of our food is cellulose. A lot of the plant material we take in consists of cellulose. And of course, that ends up as roughage. Most of that is just going to pass right through your body, but it still stimulates the walls of the intestine to produce mucus and so on. So it's not without value. We can also store additional energy as carbohydrate. So again, plants store it as starch mainly, but we store energy in glycogen. On to our next macromolecule, and this would be the proteins. Pro means first, and these are the most important molecules, I would say, if you're picking favorites. Proteins do all the work in the cell. They make up enzymes, they make up receptors, they do all sorts of things that we'll talk about in a moment. When we think about protein in our diet, we generally think about meat and legumes and so on. But keep in mind that every single cell has proteins in it. Metabolism cannot occur without protein. Movement cannot occur without protein. So what do proteins do? Well, a better question might be, what don't they do? Proteins make up enzymes, for instance, and enzymes catalyze all of the reactions that occur in your body. Those reactions wouldn't occur at all, or they would occur far too slowly to make life possible if we didn't have enzymes. So in this example here, you're seeing a disaccharide, and it's attached to this enzyme, and the enzyme is very specific. It'll only attach to that molecule because of its shape. And what it's doing is it's stressing the bond between those two disaccharides and making it more likely that the bond will break. Proteins can serve a defensive role. So antibodies that bind to foreign substances and foreign cells are made up of protein. And then they'll identify that thing as being something that should be destroyed by your immune system. Storage. So as we've mentioned, we can store energy long-term as fat. We can store it as glycogen in the liver and in muscle cells, but we can also break down proteins as well. And if you look in an egg, you've got the yolk in the middle, which is mostly lipid, and then surrounding that, the white is protein. And both of those are storage structures that store energy for the developing embryo. But we also shuttle energy around as protein in our blood. Transport proteins. So if a cell needs to move something in or move something out of a compartment or exchange stuff with its surroundings, that almost always requires a protein of some sort. It might be a protein that forms a simple channel that allows things to move down their concentration gradient, or it might be an active pump that pumps things in or out of the cell. Proteins can be signaling molecules. So in this example, we've got insulin that's being secreted by the pancreas, and the insulin will find its way into the blood, and it'll be carried by the blood to every single cell in the body. And any cell that has a receptor for insulin will respond to that signal. So it's a way of sending a message everywhere, but only the cells that have the receptor will respond. And what insulin does, of course, is it tells every cell in your body that there's more glucose than we need. You should take it in and do something with it. Store it as fat, just do something with it. Receptor proteins, so receptors for hormones like insulin, but also neuroreceptors. So here we have neurotransmitter that's being released across a synapse between a nerve cell and a cell that will respond in some way. So the receptor is going to send a response into the cell when it binds to that specific signaling molecule. Contractile and motor proteins. So there are proteins that can change shape, and they make movement possible. Movement of flagella and cilia, but also movement of your body through muscular contraction. And that's why meat, which of course is muscle, contains a lot of protein, because there are a lot of these contractile elements within the cell. And it's fascinating how this all works. We'll talk about that in Bio 112. Structural proteins. So we mentioned that in plants, cellulose provides most of the support. So the cell wall is what provides the support. We don't have a cell wall, but we do have something called an extracellular matrix. Outside of the cell, we have a layer of proteins, and they're kind of like cables or bungee cords. They're somewhat elastic, somewhat stretchy, but they support the cell. Most prevalent would be collagen. So we have lots of collagen, makes up connective tissue, but it also makes up that outer layer, that outer extracellular matrix around every cell in your body. So the take-home message here is that proteins do everything. They're even responsible for manufacturing the other macromolecules. So enzymes, remember enzymes are proteins, are responsible for making carbohydrates, and they're responsible for making DNA and RNA, and they're responsible for making lipids. Proteins are made up of amino acids. So amino acids are the building block, the monomer that makes up the protein. Each amino acid has distinct properties and characteristics, and that's determined by the functional groups. And this is the reason I wanted to look at those functional groups. There's 20 different amino acids. There's some other weird exotic ones, but there's 20 important ones. There's 20 amino acids that our DNA codes for instance, which we'll talk about later. There are some that are essential. All amino acids, all 20 are essential, but when someone says this amino acid is essential, what that means is you can only get it from your diet. So some amino acids you can make yourself, some you can't, you have to get them from your food. What happens when a protein is built is, as always, it's a process of dehydration synthesis, and we take individual amino acids and stick them together and form a bond known as a peptide. What you're seeing down the bottom there in green, that is the core of the amino acid, and that part is the same in every single amino acid. And then the part in white hanging off of that chain, that would be the functional group. Different amino acids have different functional groups. That gives each of the 20 amino acids its own particular properties. Amino acids all have the same basic structure. We've got a central carbon, and then attached to one side of that we've got a carboxyl group, and attached to the other side we have an amino group. When these are brought together to form a polypeptide, which is a long chain of these things, we always have the same orientation. So we can never have an amino group coming together with another amino group. It's always going to be an amino group coming together through dihydration synthesis with a carboxyl group. And this gives the protein an orientation, as we'll see. Now above that central carbon we have a hydrogen, and then below it you can see that we've got a variable side chain, a functional group. And there are 20 different things that could be where that R is. So that R is just representing the 20 possible things that we could see there. Note on this figure, it's showing the amino acid in its non-ionized and ionized form. Which form it takes just depends on the pH of the solution. We're not going to worry about that, but usually you'll see it represented in its ionized form. The functional group, shown here highlighted in white, is going to determine the properties of that particular amino acid. The simplest one is glycine. There's just a hydrogen there. The other ones are more complicated. Some of them are quite complicated. Tryptophan has this rather complex side chain with these cyclic structures in it. In fact, the side chain is more complicated than the actual backbone of the amino acid. But all of the ones that you're seeing here are all nonpolar. You can see that there aren't any charges on the side chains. And also we've got a lot of carbon to carbon bonds and carbon to hydrogen bonds. We don't have any nonpolar covalent bonds. So these are side chains that don't interact with water. Incidentally, I'll mention you don't need to know the names of any of these amino acids. Methionine, maybe, but we'll get to that later. I don't expect you to know the structures or the names of any of these. And also note that they can all be abbreviated, so you'll come across those abbreviations. We can also have amino acids that are hydrophilic, meaning they interact with water. If you look at the ones on the top of this diagram, none of them possesses a full charge, so none of the side groups has a plus sign or a negative sign, but we have the uneven sharing of electrons in polar covalent bonds. So you can see a few instances where we have oxygen and hydrogen bound together, or oxygen and carbon. Oxygen is more electronegative than carbon or hydrogen, so it steals the electrons away towards its own nucleus. There's others where we have nitrogen involved, and nitrogen is more electronegative than carbon and hydrogen. I should point out hydroxyl. The hydroxyl group is just written as an OH. There is actually a bond between the O and the H, but it's not shown. The one there that might be confusing you a bit is cysteine, which has that SH. There is a difference in electronegativity between the sulfur and the hydrogen, and that does set up an unequal sharing of electrons. Down the bottom of this diagram, we have the strongly polar side groups, which have a full charge. So the acidic ones have a negative charge, the basic ones have a positive charge. Basically, with the acidic groups, that amino acid, when it's placed into water, it gives up a proton and is left with a negative charge. The basic groups, they give up an electron, or they give up a hydroxyl, I should say, generally, and they're left with a positive charge. We can refer to a protein as a polypeptide. And the reason for that is when dehydration synthesis occurs, we have amino acids being stuck together, and that forms a peptide bond. Poly means many. So a polypeptide has many peptide bonds in it. Note that as we're building a polypeptide or protein, we end up with one end that has a free amino sticking out, an amino group, and at the other end we have a carboxyl group sticking out. So we call the amino end the N terminus, and we call the carboxyl end the C terminus. So any protein is going to have this orientation to it. And actually, if you know the sequence of the protein and you know where the amino end is and the carboxyl end, then you know what order things were stuck together in. So we're always adding new peptides onto the carboxyl terminus. Proteins can be remarkably complex, the most complex of the macromolecules. A polypeptide is the name for a chain of amino acids. And sometimes that's all there is in a protein, just one chain. But a lot of proteins consist of multiple polypeptides. So for instance, hemoglobin, it consists of 4 separate polypeptides, and it's not functional until those 4 pieces come together. Some proteins can consist of hundreds or even thousands of separate polypeptides. And when we talk about the structure, in order to simplify it a bit, we talk about 4 levels of structure, starting with the simplest, which is primary structure, and that is just the sequence of amino acids. Then we have secondary structure, then we have tertiary, which means third, and then quaternary, which means 4th level. And the 4th level is how different polypeptides come together. So we'll talk about each one of these levels of complexity. The primary structure just refers to the sequence of amino acids. So you could have that sequence written out on a piece of paper in front of you. That would be the primary structure of the polypeptide. So here you're seeing a sequence of amino acids, glycine, proline, etc. Note that we have an amino end and we have a carboxyl end. So every single polypeptide is going to have an orientation to it. If we change one of these amino acids, that might have a big effect on the final configuration or structure, shape of the protein. Let's say we took one of the amino acids that was nonpolar and we swapped it with the one that was polar. That would become a very different molecule, perhaps. It might fold in a different way. Now imagine that we had just a straight polypeptide and we threw it into some water. It's going to not stay just a straight polypeptide. It's going to start to fold itself, and all sorts of interesting interactions are going to occur. The first level of interactions define the secondary structure. And secondary structure is the result of hydrogen bonding, quite a lot of hydrogen bonding. So we could put a polypeptide into water and it could spiral itself into something known as an alpha helix. So a helix is a fancy term for a spiral. An alpha helix is a spiral that's formed by hydrogen bonding between adjacent amino acids. The other thing that could happen, imagine that the polypeptide bends back and forth on itself. We could get hydrogen bonding between those folds, resulting in a pleated sheet, kind of like an accordion. And this is known as a beta pleated sheet. So these are common structures that we see in all polypeptides. Note that the hydrogen bonds individually are quite weak, but there's lots of them. So we've got our polypeptide, we've looked at the primary structure and then the secondary structure, and now we're going to add another layer of complexity on top of that. So after the alpha helices and beta pleated sheets have formed, then that structure can be twisted and folded into much larger folds and loops. So this is the folding of the protein, and it's the result of a lot of different interactions. In fact, all of the interactions we've talked about. So we could have a covalent bond. That disulfide bridge that you're seeing in yellow there, that's a covalent bond between two sulfurs on two amino acids that have come close to each other. They might be far apart in the sequence, but the protein has folded itself in such a way that they've come close together. We could have hydrogen bonding, as shown in blue there. So hydrogen bonding is responsible for secondary structures we talked about, but it's also responsible for some elements of tertiary structure. We could have an ionic bond. So what if we have an acidic and basic amino acid and they come close to each other? We're going to have a plus charge and a negative charge, and that interaction will create an ionic bond. And then we can also have hydrophobic interactions. So the non-polar amino acids are going to try to basically get away from the water. They're going to be excluded from the water. All of the polar amino acids are going to interact with water. They might be on the outside of a loop, but the hydrophobic amino acids will cluster together on the inside of a loop to get away from the water. And once they do that, we can also have van der Waals forces acting between those non-polar amino acids. But keep in mind, what determines all of this is the functional groups. So the functional groups are going to determine what happens. And for the most part, it's a passive process. If you throw the protein into water, it's going to rearrange itself until it's in its most stable configuration. That's not always the case, as we'll talk about a bit later. Sometimes the protein needs a bit of help, but the protein is going to take a stable configuration. The final level of structure is quaternary structure. So there are some proteins that only consist of one polypeptide. They don't have any quaternary structure, but the vast majority of proteins do. They consist of more than one part. So you're seeing collagen here, and it consists of 3 polypeptides that are twisted around each other. And that forms a really kind of stretchy, but also quite strong cable. Then we have hemoglobin. I mentioned this before. It consists of 4 separate polypeptides, and each one of those contains a little iron atom, and that's where oxygen binds so that you can transport oxygen through your blood. So just to summarize, we've got the primary structure, which is just the actual sequence of amino acids. Then we've got alpha helices and beta pleated sheets that result from hydrogen bonding. That's the secondary structure. And in tertiary structure, we take all that and we're taking it and bending it into loops and folds. And then quaternary structure, this refers to the fact that most functional proteins consist of more than one polypeptide. So a good short answer question on a test, I might ask you to describe these 4 levels of structure and tell me the chemical bonds or the interactions that are responsible for these levels of structure. So primary structure is the result of covalent bonding. Those are peptide bonds specifically. Secondary structure is entirely the result of hydrogen bonding. And for tertiary structure and quaternary structure, every type of bond and interaction you can think of is potentially involved. So proteins have this really complex 3-dimensional structure to them. Well, what if they lose that structure? That's a process known as denaturation. So imagine here we've got a normal protein, and you can see it's quite complicated. It's got these alpha helices and beta pleated sheets. Those are the straight bits, and it's also folded into some loops. Now that can denature, which essentially means it can unravel. Now, under normal conditions in the cell, that shouldn't happen, although in some cases the protein needs a little bit of help to maintain its shape. But imagine if we had a protein and we heated it, or we changed the pH, or we changed the salt concentration. All of those things could cause the protein to change shape and even unravel. And if it unravels, we say it's denatured. The protein isn't going to work in its denatured state. So if it's an enzyme, for instance, it's got a very specific shape. There's a little notch called the active site where the substrate, the compound it's going to act on, has to bind. And if that shape changes, that enzyme can't work. In the example at the top there, you've got an egg, and remember again, the yolk consists of lipid, but the white is albumin, which is a type of protein. If you throw an egg into a frying pan, of course the egg white changes. It becomes opaque and rubbery instead of liquid. And what's happened is that albumin has denatured, it's unraveled, and that has very seriously changed the characteristics of that protein. And in the case of the egg white, you can't renature it. There's no way to make it go back to what you started with. Some proteins sometimes can be renatured. Generally in your body, you don't want your proteins to denature. And that's one of the reasons that your body goes to such lengths to maintain an internal environment. There are some parts of your body where we do want denaturation to occur, usually not of our own proteins though. In the stomach, the proteins that you eat are denatured by the very, very acidic conditions. And that means they unravel, and now those peptide bonds that were all hidden in the middle of the molecule are now out there to be picked apart by enzymes that break down the polypeptide into individual amino acids. So proteins are complicated, right? Well, it turns out they're even more complicated because they can quite often attach to other categories of macromolecules and make something brand new. So we can have lipoproteins, for instance, which are a mixture of protein and lipid. We can have glycoproteins, which are a mixture of protein and carbohydrate. They're mostly protein with a little bit of carbohydrate. We can also call these glycosylated proteins. To glycosylate something is to stick carbohydrate to it, basically stick sugar to it. We can also have proteoglycans, which are also a mixture of protein and carbohydrate, but in this case, they're mostly carbohydrate. So they're carbohydrate mainly with a little bit of protein. Glycoproteins serve a number of critical roles in our bodies and in our cells. For instance, they identify cells as belonging to you, and they identify the type of cell. They're also involved in cell-to-cell communication. What you're seeing here is a section of the plasma membrane or cell membrane, and we've got glycoproteins embedded within it. The inside of the cell is at the bottom, the outside of the cell is at the top. Notice that we've got sugar monomers attached to the outside of that glycoprotein, and there aren't any inside. Those carbohydrates that are exposed at the surface are recognized by your immune system, and that's one of the ways that the immune system knows that it shouldn't attack this cell because it's part of you. It's also the reason that you might reject a tissue graft or an organ transplant because the tissue that you're receiving has a different sequence of carbohydrates expressed on the surface. It also helps identify the nature of a cell. So for instance, during embryonic development, a cell will take cues from the cell around it when it's trying to figure out what it's going to develop into. So if we have a cell that is not differentiated or specialized yet, and it's surrounded by heart muscle cells, it will develop into a heart muscle cell, and it's getting that information from these glycoproteins in some cases. Probably the most important group of proteins are the enzymes. So as I mentioned, most of the reactions that take place within your body and within your cells wouldn't occur at all, or if they did, they would occur very, very slowly. And enzymes can speed up these reactions trillions of times. Enzymes act as catalysts. A catalyst is something that accelerates a reaction, and they can be reused over and over again. They're not destroyed during the reaction. Enzymes are quite picky when it comes to what they do. So the example at the bottom here, you've got an enzyme that's binding to a substrate. The substrate is the compound or substance that it's going to act on. And notice that there's kind of a notch in the enzyme, known as the active site, that has a very specific shape, and it will only bind a certain molecule, or just a small handful of molecules. It will bind to that substrate, and in this case, it's going to split that substrate and break off monomers. Enzymes are very large molecules, as you can see in this diagram. Every green little sphere represents an individual atom. This enzyme has a cleft, known as the active site, and that's where the substrate is going to bind, and it has to have the right shape. It starts out as a fairly loose fit, but then the enzyme will wrap around it. Once it wraps around it, there has to be a very tight fit. There has to be the appropriate alignment of charges and so on. And if that doesn't occur, then the enzyme will spit this back out because it's not the right substrate. So that's called an induced fit when the enzyme wraps around the substrate. This particular enzyme is something known as hexokinase. You don't have to worry about that, but I'll just mention that a kinase is something that will phosphorylate something. What I mean by that is it'll take a phosphate and stick it onto the substrate. So this enzyme, its job is to take a phosphate group and stick it onto glucose, which is one of the steps that occurs during the breakdown of glucose to release energy. So let's take a look at a fairly typical reaction that might be catalyzed by an enzyme. What we've got here is we've got one molecule, BC, that's going to interact with molecule A, and the B portion of that BC molecule is going to be transferred to A. Now this might happen naturally without any help, but only if those two molecules collide with each other in exactly the right way with exactly the right energy. What the enzyme will do is it will grab onto these two molecules, and it's got a very specific active site that can hold those two molecules. So the first step in this reaction is initiation. The reactants are going to bind to the active site in a specific orientation, and what's going to happen next is the enzyme is basically going to line those bits up in exactly the orientation that needs to occur in order for the reaction to occur. Then we've got what's called the transition state. Now what's happening here is the bond between B and C is being physically stressed. So the enzyme will change shape slightly. It'll put strain on that bond, and also it's bringing A close to B so that we can get a bond forming between A and B. Now what this does is it just greatly enhances the chance of the reaction occurring. It's making conditions absolutely optimal, and that lowers the activation energy, the amount of energy that's required to be put in for the reaction to occur. And now we've transferred B over to A to create AB, and that's going to be released. It's going to be released from the active site. The active site hasn't changed, the enzyme hasn't been damaged, although they do eventually wear out, but the enzyme can be reused over and over and over again. And this reaction that you just saw is one that's fairly common, where one subunit is transferred to something else. So for instance, there are kinases that I just talked about that transfer phosphates from one molecule to another. So in the process of cellular respiration, where we liberate energy from our food, what happens is phosphate groups that have been stuck to food substrates are pulled off and stuck to ADP to make ATP. There's a few reactions during the whole process where that occurs. So how might we control enzyme activity within your cell, and how does your cell control it? Well, there's two broad ways that this might occur. So let's go back to the enzyme we were talking about before. Remember that that enzyme uses glucose as its substrate, so it binds to glucose and then adds a phosphate to it. Let's imagine that we have another molecule that has a similar shape. Now that molecule might also fit into the active site. It might not fit very well, and the enzyme might spit it out right away. And even if it does bind, the enzyme probably can't do anything to it. But that other molecule is kind of an imposter. And even for the brief little period of time that it's bound to the active site, it prevents the real substrate from binding. So let's imagine a case where we have a cell and it has a tiny amount of the substrate. And then we add an overwhelming amount of the competitive inhibitor, the imposter molecule. The enzyme is going to spend all its time dealing with that competitive inhibitor, and it's pretty much going to shut down the reaction. Again, just keep in mind the competitive inhibitor isn't there for very long. Usually it's going to be released right away, but because there's so much more of it, it's going to block the reaction. There are a number of pharmaceuticals that work this way. An interesting example, I think, is what happens if you have alcohol poisoning, specifically methanol poisoning. There's an enzyme in your cells called alcohol dehydrogenase. And it's the enzyme responsible for the first in a series of steps to break down ethanol, usually. Ethanol is what we drink. It's in beer and vodka, everything else. It will also bind to methanol. Methanol is quite harmful. You should never drink methanol. Methanol is broken down into some compounds that are rather toxic, and they're especially harmful to the rods and cones of your eyes. They can cause blindness. If someone comes into the emergency room and they have consumed methanol, what they immediately do is they get the person fall down drunk. Basically, they will hook up an IV with ethanol, with vodka, essentially. They'll give them so much ethanol that the ethanol will compete for the active site on that alcohol dehydrogenase. Methanol, fortunately, is a very small molecule, and if it's floating around in your system for a while, it will be removed by your kidneys. But like I said, if it's broken down by alcohol dehydrogenase, then it becomes something rather toxic. So the goal is to overwhelm alcohol dehydrogenase until you can get the methanol out of your system. Now, the other way that we can regulate enzyme activity, and this is the method that's more commonly used by your body, is known as allosteric regulation. Allosteric literally means elsewhere, somewhere else. So in this case, we're not regulating the activity of the active site by blocking the active site. Instead, something else is happening somewhere else on the enzyme. So most enzymes will have a little cleft elsewhere that doesn't bind to the substrate. Instead, it binds to a regulatory molecule. And when the molecule binds to that spot, it changes the shape of the enzyme. Typically what happens is it causes the active site to close, and then the enzyme is shut off. There are several other factors that can control the rate of enzymatic activity. Just like any chemical reaction, if you increase the temperature, that means you increase the speed at which molecules are moving, and they bump into each other more often. So it means that reactions are going to occur more often. And if you look on the first graph there for temperature, you can see that if we start at a low temperature, 10 degrees, that would never happen in your body, of course, but let's say we're doing this with enzymes that we've extracted and we put them in a beaker. At 10 degrees, we have very little activity, but then as we increase the temperature, it gets higher and higher and higher until we get up to about 37 degrees. 37 degrees is body temperature for humans and other mammals, and our enzymes have evolved to work best at that temperature. Now, when you go above 37 degrees, somewhat surprisingly, the activity drops off very dramatically. And if you think about it, it's not that surprising because enzymes are proteins. And when we get above 40 degrees, the enzymes denature. So it's really important that you don't get too warm. If your internal body temperature gets too warm, your proteins, your enzymes, for instance, will denature and they'll stop working. There are some chaperone proteins, they're called, that can bind to enzymes and help to keep them together at higher temperatures, but even that has limitations. pH affects enzymatic activity quite a lot. We have many enzymes in our body, and they prefer different pHs. So most enzymes prefer pH around neutral, around 7. That's what we're most likely to encounter in the cytoplasm of the cell. But we have compartments within the cell, lysosomes, for instance, that are very acidic, and they contain enzymes that only function under those conditions. That's actually a way to protect the cell, because if those were to rupture and release the contents into the cell, the rest of the cell is neutral. The enzymes that are found within lysosomes are really good at breaking down pretty much everything, and they won't digest the cell. We'll talk about that later. It's kind of an interesting process. In the digestive system, the stomach is very, very acidic, and pepsin, which works in the stomach, prefers very acidic conditions. And there's some other parts of the body that are slightly alkaline as well. The substrate concentration is important as well. So in that last graph, C, what you're seeing is additional substrate being added. So we have more and more substrate being added along the x-axis. And notice that the enzymatic activity increases, or the reaction rate increases, as you would expect. There's more molecules to be involved in reactions, but we get to a saturation point. And that's the point where the enzyme is basically overwhelmed. So the enzyme is receiving inputs into the active site as fast as it can deal with them. If you want the reaction to occur faster than that saturation point, you just have to add more enzyme to your reaction. So here's a nice example of the effects of pH on the functioning of different enzymes. We've got 3 enzymes here that are very important in your digestive system. Pepsin and trypsin both break down protein, but they're found in different areas, or at least they're active in different areas. So pepsin is active in the stomach. In order for it to have its correct folding, it needs to be in a very, very acidic environment. And the stomach is incredibly acidic, less than 2, so more acidic than battery acid. Remember that the stomach is acidic so that the proteins you eat will unravel. And pepsin will take those very large molecules and break them into smaller chunks. Once the contents of the stomach empty into the intestine, the pH is going to change. The pH is going to become actually slightly alkaline in some places. Trypsin then takes over. Pepsin becomes deactivated, but trypsin has its correct folding at neutral or slightly alkaline conditions. And then it will take those chunks and break them down even further. Amylase, you can see, prefers a near neutral pH. Amylase breaks down starch. So different things can happen in different parts of the digestive system, and what enzymes are going to be active is determined by the pH. Likewise, in the cell, as I mentioned, we've got compartments within the cell, organelles. Those organelles have different pHs so that different enzymes will be active in those different areas. And if an enzyme escapes from a compartment and ends up somewhere it shouldn't, it won't do any damage. So back to our cell. If you remember, we were building a cell. That was the premise of this lecture here. And we've looked at 3 different groups of macromolecules, and these provide the cell with structure, something to eat, and also the machinery to do all the work that needs to be done. So I've represented proteins here as little lightning bolts because they do all the work. We have one last thing we have to do, and that is we have to provide the cell with blueprints that it can pass on to the next generation of cells or the next generation of individuals. To do that, of course, requires nucleic acids. Nucleic acids run the show. So DNA, found in your nucleus, is the blueprint. It's the blueprint for making proteins. RNA helps out with that. We'll talk about that next time. RNA is another nucleic acid, but DNA is a template for making proteins, and then enzymes, which are proteins, make the lipids and the carbohydrates. But we'll talk about that next class. In the meantime, here's your terminology for topic 4, which again was those first 3 macromolecules: lipids, carbohydrates, and proteins.